And so we begin actually looking at what a DNA typing profile might look like and how we analyze it. So in general, for STR testing, we have 20 specific short tandem repeats that can be found on the human genome that we are testing for. Prior to 2017, we were using 13, and recently the FBI just added another 7. The more you're typing, the better chance that you're going to get a good match, and be sure that you have the correct match. So it improves the uniqueness and the individuality of the data. So they maintain a database of probabilities for each genotype frequency. So so in the particular one for the specific STR called TH01, if you have the specific allele where you have a repeating unit 6 times, that one happens to have a probability of 0.1959. So 19% of the population has that particular allele, the 6th. Remember, everybody would have a copy of TH01. The question is it the one that's 6 long or 7 long or 10 long? And so this one happens to be a pretty popular one, a reasonable one. So you can then use the product rule, like we talked about back in chapter 2 and 3, to determine the overall probability. So if I knew all of the alleles, so I have 20 different STRs, and you get 2 copies, one from mom, one from dad, so that's 40 different alleles that we can type. So I would multiply the frequency of each one of those, the probability of each one of those 40. Note that we've observed this before. When you multiply these probabilities, the overall probability gets smaller and smaller and smaller. So I'm going to get a very tiny probability of a person having that exact identical DNA. And that's where the power of this comes in, of course. So this is what you end up getting once the profiling has been done. Remember, you're going to run a gel, and from the lengths of the bands, be able to determine which particular allele they have. And this is what the report looks like. So note up top, this is listing the specific STR. So this particular one is D3S1358. This one's TH01. And down below is showing the lengths that were found. So it looks like only 15 was found here. 6 was found at a 9.3. So there's 2 different alleles, 2730. The fact that there's only 1 peak here suggests that they had the same allele from both copies. Notice also it's like twice as tall. So that suggests that both their mom and their dad gave them the 15 allele, the one that repeats 15 times. And we go through and we have this overall version. So again, we could type this. So if I wanted to ask what is their profile for, what is their type for D5S818, oh, another one that only has 1. So this would be D5S818 11, 11, because it's just 1 peak, we assume, and you have to have 2 copies. So we assume that they're both the same length. This one would be TH01 6, 9.3. So this is how we would describe somebody's type, and then we could go about determining the probabilities by multiplying them all up. So again, don't forget about this product rule. If you multiply the individual probabilities together in decimal form, it can't be in percentage form, it has to already be converted to decimal, you will get the decimal format of the overall probability. So if each probability, so let's say that you had somebody with TH01 6, and that one I already told you was 0.1959, and TH01 9.3 is 0.2056, then we would multiply 0.1969 times 0.25056, and I'd get 0.0404. Converting that percent, that means 4% of the population would have those particular 2 alleles for this one STR. If they also had this DS profile of 1515 and this other one of 2730, and I looked up those particular frequencies, I would have the probability or frequency of this 15. I would have to multiply it twice because I got 2 copies, and then same thing for the 11 and for the 30. So I'd multiply them all together and get the total probability. Again, what is in this CODIS database? So ideally, so if you have 2 different samples, a suspect sample and a sample from the crime scene, you could match them and say these are the same DNA. But if you just have sample from a crime scene and you don't have a second sample to compare it to, it'd be nice to search a database. So CODIS has 3 different types of entries in it. So it has convicted offenders. When somebody's convicted of an offense, their DNA is collected and it goes into the database. So prior convicts would be listed in there. Also, any unsolved crime scene evidence. So in other words, maybe I go looking, I might find there was another crime scene with the same one. Also, profiles of missing persons are in there. So there's 3 types of information that's in that database. Obviously, there has to be some consent given to list it in that database, unless, convicted offenders, that their consent is because they were convicted, that automatically goes in. So there's almost 1 million profiles from unsolved cases in there. And, that number grows every day. So we can look for similar matches with the same profile in that as well.